Here when you observe any such diagram, okay, first see whether you're having a big nucleus or big vacuole blindly. So here you can observe big nucleus and small vacuoles. So definitely that's an animal cell. Well done, Aisha. Then she's the one first answered animal cell. Well done. Within 5 seconds you answered it, Aisha. Then when you go for prokaryotic and eukaryotic, pro and eukaryote basically depends on the nucleus surrounding membrane. Here you have a clear membrane around nucleus. So obviously this is the eukaryotic cell. Can't be prokaryote, isn't it? In prokaryote you won't find this kind of thread-like structure. You will have circular structure called nucleoid without any membrane, isn't it? Okay, got it, Kavya? Okay. Okay, fine, Alex. Try to answer this question. Very simple one. This is the basic question. Everybody did excellent. Yeah, correct only. All the options are correct only. Now, what will be protoplasm? One should get an idea about protoplasm. Okay. When you take protoplasm is the cytoplasm along with all cell organisms. Got it? Protoplasm is cytoplasm with all the cell organisms. That concludes the whole cell structure and function. Very happy that everyone responded. And while discussing the microorganisms, we are having 5 groups of microorganisms. Each microorganisms while I discuss, I'll touch the structure, the ultrastructure first point. Then as the second point, I'll discuss the nutrition part. Okay. Then as the third point, I'll discuss about the reproduction part. Then at last, all the microorganisms, economic importance I'll discuss. This is the order we'll follow throughout the chapter. Okay. First we'll discuss the ultrastructure. Here we discuss bacterial cell ultrastructure. Then we'll discuss the fungi ultrastructure. Then algae ultrastructure. Like that. Then nutrition. Nutrition of bacteria we'll discuss. Now. Okay. Now when you take the nutrition, most of the bacteria are not harmful. Remember this. So most of the ads and all, they give like the bacteria, the harmful one which will cause so many diseases, but no. Less than 2% of bacteria only causes diseases. Remaining all are not harmful. Okay. Now when they are causing diseases, definitely it means they are feeding on the host. But if remaining 98% are not harmful, then how the nutrition is carried out? The bacteria, most of their nutrition is saprophytic. I hope you know the meaning of saprophyte word, isn't it? What are saprophytes? Saprophytes are the one which will feed on dead matter, dead leaves or dead animal or dead plant body, something like that. On that, bacteria will feed. That's the major nutrition mode of bacteria. They are saprophytes, feeding on dead matter. Then the second category of nutrition mode in bacteria is they are parasitic. Okay, most of them are saprophytes. Remaining are parasitic. Parasitic means they will depend on a host for their nutrition. Okay, so both the methods we call as heterotrophic. This is different from autotrophic. Plants, they'll prepare their own food material called autotrophic. Whereas heterotrophic, depending on other sources, external other sources. for their nutrition. So most of the bacteria are saprophytes. Rest of the bacteria have the mode of nutrition in bacteria. And the next concept is reproduction. When you take the reproductive process of bacteria usually go for asexual mode of reproduction. Majority of bacteria will go for asexual mode of reproduction. So the term here, your asexual mode of reproduction is called binary fission. Understand the terms? Binary fission.
Yes.
Now binary here itself says you will get from a single parent 2 offspring. Fission is nothing but division of the cell. Okay, again from each individual offspring you will get few more. Like this, the division goes on. So there is no sexual interaction. This asexual mode of reproduction, which is called binary fission, happens in bacteria. Now when you take the binary fission, this is the most active dividing stage of bacteria. Okay, when you found bacteria developing very fast and you find bacterial growth very frequent, then conclude it as binary fission. So question can be asked in a way, bacteria cell rapid growth. Okay, get the word rapid growth. Rapid growth of bacteria is related to which mode of reproduction? That's the question. Which mode of reproduction? So here the mode of reproduction when bacteria goes for rapid growth is asexual mode of reproduction, and the method is called binary fission. So binary fission is nothing but from one parent you will get 2 offspring. It goes on actually dividing. This is one method of reproduction in bacteria. Then there is sexual reproduction also in bacteria. When you take sexual reproduction method, there is a method called conjugation. This is the reproduction method under sexual conditions. So here you need 2 bacteria, isn't it? So here male and female discrimination is not there in bacteria, but there is a discrimination based on the pili. Okay, so while discussing about pili, I said they are also called fimbriae, the other word. Okay, did you mention this word in the last session? Pili are also called fimbriae. Okay, now the pili or fimbriae are involved in conjugation. Assume that these are the 2 bacterial cells which are having pili on their capsule, the outermost layer of the cell, and there will be a conjugation tube formation between these 2 bacterial cells. Like this. And this conjugation tube will be kept intact by the fimbriae or pili. So pili helps in sexual reproduction. The process is called conjugation. So from here the nucleoid is there, no? That will get transferred part of it here. Isn't that this part of nucleoid get transferred to here? Clear? Can you see that? Yes, Ronak, do you have it down?
Yes, ma'am. When this binary fission happens in all bacteria?
Not in all bacteria. We'll come to understanding the classification of bacteria. Some bacteria will go for conjugation. Some bacteria will go for other sexual means of reproduction. There are processes like transmission, transduction, so many other processes are there. But at your level, this is sufficient. A sexual method, binary fission, sexual method, conjugation. The conjugation you will find frequently. Other than that, you are having some more methods also. Those are termed as transmission, transformation, transduction. So many other processes are there for the remaining bacteria. Okay, Ranak?
Yes.
Okay. Fine here. So here the point is two modes of reproduction, asexual and sexual. Asexual means binary fission. Sexual means conjugation. In conjugation, fimbriae or highly placed major role. In rapid growth, you will observe only in binary fission. That's it. That's enough. Okay. Now, I'm going to give another important point regarding bacteria. When you take bacteria have different shapes. They may be rod-shaped bacteria. They may be comma-shaped bacteria. They may be circular bacteria. And some bacteria are even spiral-shaped. So based on that, the bacteria will be given names, the terminology of bacteria. Okay, so that's all depends on the shape of the bacteria. I'm going to show the slide where you will find different names of bacteria and these shapes. So here, if you see comma-shaped bacteria, so it looks like this. This is flagella, helps in movement of the bacteria. So the shape is something like comma. Example, Vibrio cholerae. This is the bacteria name. Vibrio cholerae, which causes cholera disease. Okay. So the cholera disease causing bacteria is Vibrio cholerae and its shape is comma shape. Then you'll have a spherical shaped bacteria. Those are called cocci. Cocci is the plural and coccus is the singular. So examples for spherical shaped bacteria, Staphylococcus, singular. Streptococcus, the common cold causing bacteria, Streptococcus, bacteria name. Okay. So here, based on the shape, the terminology is differentiated. So Vibrio cholerae comma-shaped bacteria, Staphylococcus, Streptococcus are spherical bacteria. Then, if you have rod-shaped structures of bacteria, then they are called bacilli. Bacilli is plural and bacillus is singular. So the rod-shaped bacteria, best example, E. coli. Then Salmonella. These two are the bacteria which are rod-shaped. Then spiral type shaped bacteria. Examples, Treponema borrelia. See students, why I am giving the bacteria name is, you may get a question like identify the prokaryotes from the given. Like that you may have a question. Identify the prokaryotes from the given list. They may give a list like Treponema, Salmonella, Vibrio cholerae, something like that. And we have to identify the bacteria name. So doubt. How we come to know whether the particular bacteria is rod-shaped or spherical shape, isn't it? How one comes to know? There should be some process where we can observe it under microscope and tell that this is Streptococcus, this is Vibrio cholerae, something like that. So on that background, I just want to give an account of how here they have used words magnification of bacterial cells under microscope. Okay. Usually we will observe under compound microscope. You are having electron in simple microscope also, but in school lab use simple microscope or its compound microscope based on the category of school, CBSE or state syllabus. Magnification is something related to resolution. How much in detail you will observe anything under microscope is magnification. Okay. Now here, when you take the bacteria can be observed under microscope if only they are stained. Just by taking a drop of water and observing under microscope will not give you any idea about the bacteria. Okay, it has the process of staining is called Grams staining. What happens here is, I'll give how the whole practical will be taken up. In your lab of biology, if you ever come across with a thing called glass slide, have you ever been to biology lab? Anyone of you? Okay, Tavia, you have been. Ronak, you also. Okay, fine. School bio lab. Okay, yeah. Yeah, Naisha. You have been? Okay, fine. So, glass slide, a thin slide of glass looks like this. Okay, if probably pandemic gets sent for the at least next academic session, you would be 9th standard. When you go to your school biology lab, at least in the next academic session, try to do this practical under supervision of any of your lab assistant. Okay, take a glass slide, just add a water droplet on that. Just a droplet. Now take the glass slide onto a Bunsen burner. Lit the Bunsen burner. Just upon the Bunsen burner flame, keep this slide, move forth and back like this. Okay, just for a minute. This water drop will dry and will get stick to the glass slide. This is called smear formation. Smear. Lab word, biology lab word. Okay, smear is nothing but the water drop you had taken on the glass slide now that gets fixed. Gets fixed on the glass slide when you kept on the Bunsen burner flame to and fro. Just once. Once you got a smear, means glass slide becomes fixed water drop. Now there are 3 steps you have to follow. Every biology lab or even you go for chemistry lab, you will find these chemicals. The first chemical is crystal violet. Add a drop of crystal violet onto this. Then keep the slide under running tap water. Wash the crystal violet solution. Then add ethyl alcohol. You will find ethyl alcohol everywhere. Or you can add acetone for that case. Just add 1 drop or 2 drops of acetone on the slide. Wash it off again under running tap water. Then add saffron. Okay. And again wash it away. So you have done 3 times washing. Even then you will find the smear intact on the glass slide because you fixed it now. You fixed it on Bunsen flame. So the smear will not get rid of. It will fix to the glass slide. First add crystal violet, wash it off. Add ethyl alcohol, wash it off. Then add saffron, wash it off. Now take a tissue paper. Just put the tissue paper on the glass slide. Don't wipe it off. Just put it on and take away the tissue. The tissue paper, just put it on and take away. Now observe this glass slide under microscope. Compound or simple, anything is okay. Now this is how you can observe bacteria. Here the bacteria randomly we are calling, they may be in rod shaped. The black and white pic I'm showing left side of the slide. Or you may have the round shaped bacteria, something like this. Isn't it? Either they may be this shape or comma shape, something like this we are studying. But you can really observe them under microscope if you follow Graham's staining method. Staining is nothing but you are adding different colors, that's all. 1, 2, 3 colors you are adding. Okay, and Graham is the scientist who contributed to this process. Now then, after Graham's staining, if you observe this slide, under compound and simple microscope, you may get images either like A or like B. Now see, in image A, you can observe circular bacteria and rod-shaped bacteria also. Can you see those things? They are almost in bright red. You may get this observation under microscope, or you may get this observation, the slide B I'm showing. So they are dark purple violet, isn't it? The color difference you can observe. Purple violet. Here also you can observe rod-shaped bacteria numerously. And some are circular bacteria. Okay, this is how you can look under microscope if you stain on a glass slide. You just took a water drop and these many bacteria you can observe. Just fresh water drop. So here, these bright red color bacteria are called gram-positive bacteria. Positive. And dark purple bacteria are called gram-negative bacteria. Gram-negative. So bacteria can be broadly classified not only based on their shape. This is the point you have to understand. Based on the staining technique also. Based on the staining technique also, bacteria can be classified as gram-positive and gram-negative. Clear? So from gram-positive thing, you can say the gram-positive bacteria are the one which have taken the counter stain, safranin. That's why they are in red color. Gram-negative bacteria, they have taken the crystal violet color. That's all. Okay? Clear with the concept? Any doubt? Why I took this much time to explain the whole gram staining is, a question can be asked, bacterial classification is based on, you may have option, first option, shape of bacteria. Second option, staining process. Third option, based on the presence or absence of some cell organelle they will give. And option D, they will give all the above. You will get stuck with the first option. Because everybody explains shape of bacteria helps in classifying bacteria. Rod shape, something like that, isn't it? But staining process is also helping in. Ronak, you want to take notes? Okay, I'll display the thing, no problem. So staining process is also the one which will help the bacteria classification as gram positive and gram negative. Okay,